Good morning, dear students. Today we are going to learn about compound lipids. In the former videos of this chapter, we learned about simple lipids like triglycerides, its properties, everything. So today we are going to see about compound lipids. Compound means something is there as additional. So let's see the definition. Like simple lipids, they are also esters of fatty acid with alcohol. Esters of fatty acid with alcohol. We know that esters are only forming when an acid combines with alcohol. So that is there. Along with that, present some additional functional group that make that lipid compound. So compound lipids are, again, they are esters of fatty acids and alcohol, and they possess some additional functional group. Okay. Some examples for compound lipids are cerebrosides, glycolipids, sulfolipids, etc. How can we classify these compound lipids? Mainly as phospholipids and glycolipids. And first, we are going to see about the phospholipids. This phospholipids contains what all the its composition, glycerol will be there, fatty acids will be there, and a nitrogenous base also will be there. So that is about its composition. And these phospholipids, they are what's this another importance? They are the major lipid constituent of cell membrane. So that's another feature. Which is the simplest phospholipid? Simplest phospholipid. It is phosphatidase. Simplest phospholipid is phosphatidase. And these phosphatidase are actually, they are derivatives of phosphatidic acid. They are derivatives of phosphatidic acid. And this phosphatidic acid, again, it's the simplest phospholipid. Okay, so simplest phospholipid is phosphatidates. They are derivatives of phosphatidic acid. Okay, then what about the composition of this phosphatidates? They are composed of uh, glycerol, 3-carbon compound glycerol. And to that glycerol, 2 fatty acid chains are attached to carbon 1 and carbon 2. And the third carbon, this third carbon is esterified to one phosphoric acid. Phosphoric acid. So that's all about phosphatidates. So let's see some other features, importance, properties of these phosphatidates. These phosphatidates are the smallest, simplest phospholipid. As it is the simplest form, they can act as the precursor for complex alcohol containing phospholipids. Okay. Next point is that this phosphatidate is a strong anion. Anion means we know that it's a negatively charged. As it is a strong anion, it contributes negative charge density to the membrane. As it is negative, it can give negativity to the membrane. And there's one enzyme called lipid phosphate phosphohydrolases. Lipid phosphate phosphohydrolases. This is the enzyme. What's the role of this enzyme? Because in our body, this phosphatidate should be always kept in a very low level. So whenever its level becomes high or increases, that time this lipid phosphate phosphohydrolases, it convert that phosphatidic acid to diacylglycerol or DAG. And this diacylglycerol, we know that it is a metabolic precursor. It's a metabolic substrate or precursor in the metabolic pathway of lipids. So, as this phosphatidic acid get converted to diacylglycerol, it can be easily metabolized to its finer, simpler forms. By that, we can maintain the low level of phosphatidic acid inside these cells. So, this is a regulatory method. Just note the name of that enzyme, lipid phosphate phosphohydrolases. And also this phosphatidate it has an asymmetric carbon. That structure we have seen that one carbon only is asymmetric. We know that asymmetric carbon means all its four valency should be means bonded with the different groups. Then only we can call that carbon as asymmetric. So it possesses one asymmetric carbon, and because of that property, it can exhibit optical isomerism. Optical isomerism also we learned in the carbohydrate chapter. The ability to change the uh, direction of movement of plain polarized light. That is optical isomerism. We have learned that in that chapter. I am not going to explain that here. And as optical isomerism can be shown by this phospholipid, we should know that which isomer is found in nature. It's L isomer. Isomer means that isomer which can turn or shift that plane polarized light to the left side. So L isomer of the phosphatidic acid is present found in the nature. Then again, we are moving to some properties of this phospholipid. So till here, I'm talking to you some points about phosphatidic acid. Now again, we are going back to phospholipids in general, its properties. Generally, what are its other properties? They're amphipathic. We know that what is amphipathic. Amphipathic means any molecule that have a polar part and a non-polar part. Such molecules are called amphipathic. So phospholipids, it possesses polar or hydrophilic part and a non-polar hydrophobic. In the phospholipid, the fatty acid chains are the non-polar. Glycerol and phosphoric acid, they form the polar part. And also they can form micellae. What's micellae? Micellae. You might have been learned this term in some other topics before. Anyway, they are certain means molecular aggregates. They are certain molecular aggregates. How it formed? When the phospholipids it is distributed in water, they assume in the form of a sphere by keeping their hydrophobic parts away from water. So in this way, it assumes the shape of a sphere, an aggregate. So that aggregate is called micellae. So the phospholipid has the property to form micellae when distributed in water. Okay, next I'm going to show the image of that micellae. This is the micellae. And the micellae formation property of the phospholipid helps the easy digestion of lipids. How? Because as the complex lipids get broken down to small, discrete particles, because of the property to form micellae, it prevents the further aggregate formation of lipids. So that is an important property by this phospholipid to speed to fasten its digestion and also its absorption. Okay. Next, another property, it can form liposomes. What are liposomes? Liposomes are very simple structures. They are again they are spherical shaped vesicle. Vesicle means it can carry something, and it is composed of phospholipid bilayers. We know that bilayers means something like in this way. So it can form phospholipid bilayer in the form of a sphere. Sorry, entirely this is forming, and it will form in the form of a sphere, and there will be some space inside. Here, there will be some space. Uh, and this bilayer closely resembles the structure of cell membrane. And the ability, this structure is called liposome. And the ability of this liposome is that it can encapsulate, it can carry hydrophilic or lipophilic drugs inside, and allow this vesicle to become a useful weapon, useful tool for drug delivery. So that is the importance. I'm just going to show you an image of liposome. Here, this is one liposome. This is the lipid bilayer, and inside we can see the drug. It is in a crystallized form, and also lipid soluble drugs can be kept here. So in that way, we can deliver a drug to the body of a patient with the help of the liposomes. So a liposome is a drug delivery tool. So phospholipids has the ability or the property to form liposomes. So for your exam, if a question came like that, what are liposomes? Then you need to give this definition and also its application. So that's why liposomes have important applications in cancer, chemotherapy, antimicrobial therapy, gene therapy, vaccines, and diagnostic imaging, because it can carry something inside in it and can deliver to the target points, target sites inside our body. Then another property, it can form accusomes. What are accusomes? Accusome is almost, again, it is a spherical structure, but they are nanoparticulate carrier systems. They are nanoparticulate carrier. Nanoparticle means we know that very small particles. Carrier system. And it has a three-layered self-assembled structures. These accusomes, they are nanoparticulate carrier system. They are also carrier in function. They are also using in drug delivery, and they are having three layer. In that spherical structure, three layers are there. And what all form the three layers, or what each layer? That way I am going to tell you. The innermost layer, that is of a solid nanocrystalline core, means it is composed of means small, so many nanoparticles as a filling. It forms the core. Okay, and to that present a coating of polyhydroxy oligomers, a carbohydrate coating, polyhydroxy oligomers. And to its surface, we are giving the drug. So something, whatever thing we need to carry or to deliver, that we are giving, that substance is giving us the third layer as a coating. It's adsorbed to that carbohydrate layer, oligomer layer. So in that way, this also can carry drugs. Before that, I will show you the structure. This is exosomes. Here we can see the drug adsorbed over that oligomer coat, and inside this, that nanoparticulate core, it gives stability, and this oligomer coat gives stability for or it stabilizes the drug adsorbed to the surface of this coat. So that is the exosomes. And this delivery system, it is successfully utilized for the delivery of insulin, hemoglobin, and various antigens. And also it is used for the oral delivery of certain enzymes like ceratopeptidase. So these are all its applications. Application of aqueous forms. Then another property of phospholipid is that it can form biomembranes. How? By aligning themselves to form monolays. Aligning themselves as monolays with their polar heads pointing in one direction and the non-polar tails in the opposite direction. And also it can form bilays. So bilays, how these bilays are forming? Because of the hydrophobic interaction. Through hydrophobic interaction, they can form bilays also. These are all the properties. So I hope that you got a general idea about phospholipids. And by that way, today I'm going to wind up our section. In the next class, next video, we will start to see about different types of phospholipids. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.